Thank you, Joe. So I'd like to thank Joe and Julie and Karen for putting on this conference. I wasn't able to attend the one 2 years ago, but I think this is a great thing. I work with the Dravet Syndrome Foundation and some of the SCN8A groups, and I think it's really good bringing clinicians and scientists and families together to talk about the disorder. So I'm going to tell you about some of our work. I believe this is my using patient-derived cells to model epilepsy. So you've heard a lot about different epilepsy genes. So we're getting better at discovering epilepsy genes, especially with exome sequencing, so-called next-generation sequencing. And we make animal models to test hypotheses about how genetic abnormalities lead to epilepsy. And then there are a variety of model systems, and you've heard about fish models and mouse models. There are also potentially worm models and fly models of epilepsy. And I'm going to focus mainly on patient-induced pluripotent stem cell models, as well as a little bit of a rodent model we're trying to develop. So how does one study a gene mutation that affects an ion channel or another neuronal molecule to cause a seizure phenotype? And a lot of times we can express the mutant human gene in what we call heterologous systems, like frog eggs or human embryonic kidney cells. And we use these cells because it's easy to express the genes in these cell types. And you can, these are really big cells. It's easy to do electrical recordings from them. And they give us some information on certain properties of the molecule, like for ion channels, the biophysical properties, but they're not brain cells, so they only give you a limited amount of information. So scientists turn to making animal models, and they can make transgenic mice that express mutant human genes that cause epilepsy, and then study the mice, see if they have seizures, and you've heard a little bit about that. And we've taken the approach, we and others, of taking patient cells and making patient-specific neurons or brain cells in the dish to study them and see how they cause seizures and potentially come up with personalized therapies. So what is a pluripotent stem cell? So it's actually pretty simple. There's only two things that make something a pluripotent stem cell. It can self-renew, so it can make more of itself by dividing, and it can give rise to all of the cells of the organism. So the classic is the embryonic stem cell that comes from the inner cell mass of the blastocyst, a developing embryo. And if you take it out and culture it in the dish, they make more cells, and they make all three germ layers: ectoderm, which is neurons and skin; mesoderm, like blood cells; and endoderm. So today I'm going to give you some more background on induced pluripotent stem cells, talk about what we learned studying sodium channel mutations that cause epilepsy in patient-derived cell models, Dravet syndrome and SCN8A associated epilepsy, and then talk about our work with PCDH19 epilepsy, and end with kind of a glimpse into the future where we can make mini brains in a dish. So what is the induced pluripotent stem cell method? Well, it was pioneered by Shinya Yamanaka, a Japanese scientist who also has a lab here at UCSF at the Gladstone. So in 2006, he figured out how to take a mature adult cell from a mouse, like a skin cell, and using specific developmental genes that he forced the expression in those cells, he could reprogram it to a pluripotent stem cell that could then give rise to all different kinds of cells. A year later, in 2007, his group and Jamie Thompson's in Wisconsin figured out how to do it in humans. And the importance of this approach was quickly recognized, and within 6 years, he was already sharing a Nobel Prize for this discovery. It's a really powerful approach. So you take cells from adult organism like a mouse or a person, you use transcription factors like these genes to express them, and they reprogram these cells into these stem cells. You grow the colonies, and in theory, you can give them back to patients after making them into whatever tissue the patient needs for regenerative therapy. Or you can study it in the dish and test disease mechanisms screen for toxicity and also look at therapies in the dish. So there are a variety of different methods to generate induced pluripotent stem cells. The original method was using retroviral vectors, and that's what Shinya Yamanaka used. And that was important because these are developmental factors. And the reason retrovirus worked well is because the retrovirus gets silenced, and you want to eventually turn off these developmental factors and let the cells mature and differentiate. So that worked, but retroviruses integrate into the genome and can cause problems, can be oncogenic, cause cancer. So other methods came around, and now most people either pay Invitrogen a lot of money to get Sendai virus kits to reprogram, or we can use episomal plasmid vectors and transfect. And the efficiencies vary, and there's some advantages and disadvantages, but we're getting better at doing this. Our lab still uses fibroblasts or skin cells, but a lot of groups are doing it with blood cells now, and the field's moving that way. So what's nice about studying brain diseases with these induced pluripotent stem cells is there are a lot of different cocktails that people have figured out in order to, once we reprogram a skin cell to a pluripotent stem cell, to make different types of brain cells to study. And that's important. So we can make iPS cells into hippocampal-like progenitors and then dentate granule cells. Rusty Gage has a protocol to make CA3 pyramidal cells, so we can study hippocampal neurons. We can make medial ganglionic eminence-like progenitors, which are the interneuron progenitors, and make cortical interneurons. We can make dorsal progenitors and excitatory neurons, and different types of glia, astrocytes and oligodendrocytes. There's a more recent approach that Thomas Sudoff has pioneered, where you can get neurons. So these are kind of laborious and expensive techniques. You have to culture them with a lot of growth factors. It can take many weeks. And by using this transcription factor, neurogenin 2, with an inducible system, Tom Sudoff showed that you can go from embryonic stem cells or iPS cells to neurons within just a couple weeks. So it speeds up the process. It only gives you excitatory neurons, so you don't get interneurons, but it gives you a homogeneous population to study. So there's some advantages to that approach. We're starting to work with that, but the data I'll show you today is with the classic differentiation protocols. All right, so again, we go from skin cells to brain cells. And what's nice, you heard about CRISPR gene editing a little bit in some of the earlier talks. With stem cells, it's an important approach that adds to the power of the stem cell approach, because you can make what's called isogenic control. So if you have a patient's cells with a mutation, usually we compare it to other controls, but they differ in a lot of different genes in terms of their genetic background. But if you correct the patient's own mutation, you have exactly the same cells that only differ by the presence and absence of the mutation. So it's a really powerful approach. If you can't get access to patient material, you can actually take a control line and induce a mutation to make a virtual patient that you compare to the control line. So these are making the approach even more powerful. Okay. So I'm going to talk a little bit about Dravet syndrome, because this was the first disease we started studying with induced pluripotent stem cells. And you've heard a bit about Dravet syndrome. So it's an epileptic encephalopathy, typically comes on in the first year of life. And it's in over 80% of the cases caused by de novo mutations in the SCN1A gene, which encodes a voltage-gated sodium channel. And the mutations can occur anywhere in the gene and protein, and it's thought to be what we call haploinsufficiency, which is loss of function. So it's a de novo dominant mutation, so you have one normal copy and one mutant copy, and so you make only 50% of your normal sodium channel. But we don't understand why that causes seizures and intellectual disability and other symptoms. So Bill Catterall, an investigator at the University of Washington, kind of started the early work with a mouse model to study Dravet syndrome. And intuitively, it doesn't make sense. So sodium channels are responsible for action potentials. And to have seizures, you have too many action potentials. So if you lose your action potentials from less sodium current, then you shouldn't have epilepsy. So that was kind of the counterintuitive. And Bill Catterall's hypothesis was that, well, if you lose it just in the interneurons, then that can cause disinhibition and seizures. And he showed by recording from hippocampal cells from this knockout mouse model that the excitatory pyramidal cells had normal current, but the heterozygous and homozygous knockout inhibitory bipolar cells had decreased sodium current, supporting the disinhibition hypothesis. It's probably a little more complicated than that, which I'll get into a little bit, but we wanted to test this interneuron hypothesis using patient-derived neurons. So in 2009, Yu Lu, a postdoc in the lab, reprogrammed fibroblasts from 2 Dravet patients with the Yamanaka factors in retroviral vectors. And so you start with fibroblasts. Within a month, you get nice pluripotent stem cell colonies, which look just like human embryonic stem cell colonies. They express pluripotency markers like OCT4, SSCA4, NANOG, alpha phosphatase, and you can differentiate them through embryoid bodies into all 3 germ layers. So we showed that we can make teratomas in immunocompromised mice. So Yu Lu differentiated them into neural progenitor cells. So they might make these nice neural rosette-like structures that express stem cell markers, PAC6 and Musashi. He can expand them as neurospheres and differentiate them into neurons, and they make beautiful neurons. With this protocol, about 80 to 90% were GABAergic neurons. About 5 to 10% were these big, plump, excitatory neurons that didn't express GABA, but they expressed the vesicular glutamate transporter. So we gave these cells to our collaborator in Laurie Isom's lab, Luis Lopez Santiago, and he recorded from pyramidal shaped cells and bipolar shaped cells to test Catterall's hypothesis, and got nice sodium currents. And to our surprise, compared to 3 different control lines, instead of seeing decreased sodium current, which would be upward, we saw increased sodium current in both of the patients, in both pyramidal shaped and bipolar shaped cells, which isn't what we expected. Another talented electrophysiologist in Laurie's lab, Yukun Yuan, recorded in current clamp from the cells, and compared to the 3 controls lines, the 2 patients had a lower threshold for action potential initiation, and with stimulation they had more repetitive firing, and they had lots more spontaneous activity compared to the control. So this was, a rewarding finding that we wanted to see. It was our epilepsy in a dish model, and it kind of validated the model. And long story short, we have some evidence that we think loss of one sodium channel leads to increased sodium current because we think there's overcompensation by a different sodium channel, SCN8A, which encodes for NAV1.6. And supporting that, a different mouse model, also a knockout mouse made by Jennifer Kearney and Al George, in 2-week-old mice, they see the same decreased current in interneurons, but if they look a week later at a P21 to 24 mouse, they see increased current in excitatory neurons. And it depends on background. And that's one of the difficult issues that we don't like to talk about when we work on mice, but the background makes a lot of effect. In some models, mice will have seizures with a mutation on one background and no seizures on another background. But she also showed that these excitatory neurons had increased bursting and spike activity, and there was a significant but small increase in SCN8A in these neurons. So it supported an overcompensation by this other sodium channel subunit. So I think the story may be more complicated than just the interneuron hypothesis lets on. All right, so that was our initial work on Dravet syndrome, and then we moved to a different protocol to make excitatory cortical neurons from iPS cells. This is a dual SMAD inhibitor protocol. We get about 95% excitatory neurons with this protocol. As shown here, these express the transcription factor CTIP2 in the nucleus and a neuronal marker TUJ1 in the cytoplasm. And this is a transcription factor expressed by deep layer projection cortical neurons. We can give reporters. We have lentiviruses that express CAMK2 alpha expressing GFP, driving GFP or RFP, which labels excitatory pyramidal neurons, and we can mix them and record from them prospectively, so it helps the electrophysiologist. And so we use this approach to study a different epilepsy caused by SCN8A mutations, which encodes NAV1.6. And unlike Dravet syndrome, which are loss of function mutations, these are point mutations, single nucleotide substitutions, that probably cause a gain of function in this protein. And so Miriam Meisler made a mouse model by introducing this mutation with talons, and we recorded from the mice and showed that they had spontaneous seizures. And Laurie Isom's lab did electrophysiology and showed that they had an increase in persistent sodium current, which had been found for some of the mutations in heterologous systems. So we wanted to test it in patient-derived neurons, and so we have two patients, one with a substitution here in the carboxy terminus and one in this transmembrane region. Both have severe epilepsy with intellectual disability. And Andrew Tidball is the postdoc in the lab who generated the iPS cells and differentiated them and gave them to Luis and Laurie's lab. And compared to the control, when we look at transient sodium current, which is this big spike, they were not different compared to the control. But when we look at persistent sodium current, which is this smaller change that goes on longer, it was increased in the patients compared to the control. And it's quantified here. There was a trend toward decreased transient current in the patient, but no change in persistence, so the percentage of persistent current was higher in the patient, just like what we saw in the mouse model. So now we have a phenotype. What we want to do is be able to test drugs and see if we can find specific drugs that work. And so the question is, how do we do it in a rapid manner? For this, we've turned to a company, Axion, that makes these multi-electrode arrays. And you heard in Ann Paduri's talk a multi-electrode array that they put on the fish. Well, this is one that you can culture cells in, but what's nice is it comes in a multi-well format. 12 well plates that have 64 electrodes per well, 48 well plates that have 16 electrodes, and 96 well plates that have 8 electrodes per well. And so you can record, and the spikes look like this in these raster plots. We convinced ourselves they were action potentials by giving tetrodotoxin, and that blocks them. You can see them as a heat map. And I don't think this is going to work, but I'm prepared because we have it here. And this is what it looks like, hopefully, in real time. And so these top two rows are controls, and these are from the patients. And these are all action potentials from the iPS cells that we've cultured on the plate. So now we can add drugs, and we can add different drugs to the different wells. Okay, get rid of that. And what these plots show, this is percent inhibition of spontaneous activity. And we gave two drugs, sodium channel blockers, that are known to work well in SCN8A epilepsy. So again, it's different than Dravet syndrome, where we wouldn't use sodium channel blockers. And red is the patient, and blue is the control. And we see that oxcarbazepine, I spelled it wrong, but oxcarbazepine works for both, but more so in the patient. Lamotrigine works for both. Levetiracetam, which doesn't work in SCN8A patients, doesn't have a good effect on either. We tried, for the heck of it, riluzole, which is actually an ALS, the Lou Gehrig's disease drug, but it also blocks persistent sodium current, and actually that worked better in the control than the patient to decrease activity. But what we learned is that just looking at firing rate is probably not the best measure of activity and epileptic-like phenotype in these cells. So we look at things like bursting, and we find compared to the control, the patient has increased spikes per burst and increased burst duration. And when we give a drug and look at these measures, it has little effect on control in terms of spike per burst or duration, but it brings the patient cells back toward baseline. So we're looking for something that doesn't affect the control neurons, but that normalizes the patient-derived neurons. And I think this is going to be useful in terms of validating the model by showing that it works in the same way we expect patients to respond, and also discovering new drugs. Okay, so now on to what you guys are most interested in, PCDH19 epilepsy, which unfortunately is a little harder to study with induced pluripotent stem cells. So we started, and this was an MSTP student in the lab, CC Du. She reprogrammed fibroblasts from 2 patients, one with A to C transversion, one with a large deletion. And I'm only showing you cells from patient 1 because patient 2 had no phenotype, because with the large deletion there was preferential X inactivation of the mutant copy. And that's something that Ricardo Dolmesh and Joseph Getz has seen with their IPS cells. So if the disturbance is too bad, they preferentially turn off the bad copy and you don't get a phenotype when you study them. But with this one, we found evidence of both cells. These colonies don't look as nice as the earlier ones because they're on feeder-free. The earlier ones were grown on mouse embryonic fibroblasts. So these are feeder-free, reprogrammed with episomal vectors, differentiated with dual SMAD approach to make cortical neurons that express double cortin and MAP2AB. And then the other thing that's difficult with PCDH19, you heard about the X inactivation. So with IPS cells, the literature is kind of iffy about whether there's normal reactivation and inactivation of the X chromosome. So we worked with a talented human geneticist, Sundeep Kalantri, who helped us do fluorescence in situ hybridization for EXIST and staining for trimethyl H3K27 to show that there was inactivation in neurons, and you only express 1 copy. Kevin Egan shown if you passage your IPS cells too long, you start losing the inactivation and you get a gene dosage problem. So that's something to worry about. But CC was able to show that both with sequencing that the mutation was expressed in the cells that she got, and she saw some semblance of a phenotype, but we're still working it out. So she stained for PCDH19. As you've heard, these antibodies aren't that great. There may be a difference in the staining pattern, but I'm not hanging my hat on that. What she did show that we're pretty confident about was when they form these neural rosette-like structures, which are neural tube-like structures, they're supposed to make a tight apical membrane that you can label with PKC gamma and N-cadherin. And with the patient cells, we see disruption of that. And that's been seen with other disease-causing, autism-causing mutations. CC differentiated the cells into neurons. There may be a subtle dendritic phenotype when she stained with MAP2AB. We're still working on that. When we look at transcription factor expression, this is TBR1, a deep layer marker in green, and TUJ1 in red. It seems to be a bit dysregulated. And when we look at CTIP2, a deep layer, and SATB2, a superficial cortical layer marker that comes on later, we don't see it yet in the control, but we see it everywhere in the patient. I don't really understand why these transcription factors would be misexpressed. And again, this is only 1 patient, and we're still working this out. But because we don't know what percentage of the cells are turning off the mutant copy and what are turning off the wild type, we wanted to take a different approach. Actually, I'm jumping ahead of myself. So those were excitatory neurons. We also wanted to make interneurons to be able to study. We used a protocol from Suchun Zhang that makes medial ganglionic eminence-like interneuron progenitors and then interneurons. And CC was able to get over 90% GABA-positive interneurons in these cultures. We recorded from them, and they fire with spike frequency adaptation like interneurons. So unfortunately, they take many months to grow the cells, but it's a good protocol for making interneurons. So we looked at control versus the patient, and we seem to see a fasciculation defect. So this is GABA in green and MAP2AB in red, and we see normal processes in the control, and we see these big hyperfasciculated processes in the patient interneurons. The other thing we see in the interneurons, MGE progenitors are very migratory, as others have shown earlier today. They migrate from the medial ganglionic eminence all the way to the cortex. And so when we look at the control cultures, there are small clusters, but most of the cells are spread out. When we look at the patient, they seem to persist in these big clusters. You can also see the hyperfasciculation that's not present in the control. So That we feel is more consistent, the interneuron phenotype, than what we see with the excitatory neurons, but we're still working on it. In order to get around the problems of exon activation, more recently we've been performing mixing experiments, and you've heard a little bit about that with Paul's talk, but I'll take you through it. So what we're doing is we're labeling cells green and red with different fluorescent recorders. So we take control cells that we label green and red, we mix them, and we expect a normal outcome. We take unaffected male, so father of a patient with a PCDH19 mutation. We label half the cells green, half the cells red, mix them, we expect it to be normal. But when we mix the control in green and the unaffected male in red, we expect that to have an effect on development. And the father has a 14 base pair deletion that causes a frame shift that leads to a nonsense mutation. We haven't confirmed lack of expression, but that's what we expect to find. So here's what happens. We're mixing at the neuroprogenitor cell stage, and in the control in green and red, they're mixing. They have process outgrowth. The unaffected male, green and red together, again have mixing and process outgrowth. But when we mix the control and unaffected male, it seems like these cells want to stay together. The processes look strange, and many of them are not growing out of the culture. You can see here with double cortin labeling, they're just staying within this conglomeration rather than growing out. Here's another example. Control and control seems normal. Unaffected male with unaffected male seems normal. But we get these curvy, strange processes, and the cells seem to want to stay together. So it's early, it's preliminary, but it resembles what maybe Paul is seeing with the mouse model. So we're excited about this. We also are starting to do it with the interneuron cultures. And eventually we want to mix the two, interneuron and excitatory neuron, together. Okay, so this is a work in progress, our attempt at an animal model of PCDH19 by making a mosaic with in utero electroporation. And Maria brought up and introduced this technique to you. So you take embryos in the uterus, you inject your DNA into the ventricle, electroporate across, and get them into the dorsal progenitors and let them develop, and then look, and if you label with a fluorescent protein, you get these nice pyramidal cells that are labeled over time. And you can express a mutant gene, or what we're doing is we're using CRISPR to knock out PCDH19 in the embryonic forebrain. And we're doing it in rat. And this is work by Yu Wang, a postdoc in the lab. So basically, he's injecting the CRISPR at E14.5, the guide RNAs with Cas9, and also a fluorescent label. And he's looked in vivo, and at first we thought we were seeing a migration phenotype, and we did more animals, didn't see a migration phenotype. At first he thought he saw a dendritic phenotype. That didn't pan out. What we think is happening in vivo is at E14.5, we get it in the radioglial cells, but then it takes a couple days for the Cas9 to start working, and we think it's too late, we missed the boat. And what Paul showed suggests we need even earlier if it's affecting E910 radioglia in mice. So that's been a challenge. With this approach, he electroporated in vivo and then cultured the cells in vitro for 10 days and sees a dendritic outgrowth phenotype that we're still trying to, he still needs to replicate. We're pretty convinced we're getting good knockout. So there's no good antibody to show that we're knocking out the gene, but we sent it for targeted sequencing, and about 70% of cells show knockout of the gene. So, but with the fluorescent protein, we don't know if it's the 70% cell that we're looking at or one of the 30% group. So this approach is a little complicated. It also only gets the excitatory neurons. We're targeting interneurons. As you heard with some of the questions earlier, it's hard to target these medial ganglionic eminence interneurons, but you can change the polarity of your electroporation, get some of them, and here we have a DLX56 reporter, and the cells co-express GABA. The problem is we want to get parvalbumin interneurons, but they develop even later at a time when you can't really electroporate and get surviving embryos. So our plan is to use viral vectors to try to knock it out in interneurons this way, but we're not there yet. All right, so in the last part of my talk, kind of where our group and other groups are heading for the future. So it's kind of like science fiction, but it's here. So I've been talking about growing human-induced pluripotent stem cells in two-dimensional culture in a dish and looking at them, and they can form networks in a dish and make synapses on each other, but it's two-dimensional. It's not like the brain, and three-dimensional, and we're talking about epilepsy, which is a network disease. So how do we do three-dimensional cultures? Well, it turns out if you take human-induced pluripotent stem cells and just grow them in suspension, floating in media, they self-organize to form mini brain-like structures, cerebral structures. And Noblick and colleagues showed this several years ago. So they grew them in the dish for several months, and you get these large 2 to 4 millimeter structures that have progenitor layer here expressing SOX2 and a neuronal layer on the outside that expresses neuron-specific beta tubulin. They express forebrain markers like FOXG1. You get ventricular-like structures with choroid plexus, putatively making cerebrospinal fluid. You can get hippocampal onlaga. They see retinal-like structures, kind of freaky to make an eye in the dish. Here is a ventricular zone that expresses SOX2 and phosphovomentin, where you see the cells dividing where they should be at the lumen of the ventricle, and an outer subventricular zone. And here you see several cortical layers, a deep cortical layer that makes CTIP2 and a superficial cortical layer that makes SATB2. So this approach came out, and I had to twist Cece's arm to try it, but she eventually did. And she got these big structures that were anywhere from 1 to 4 millimeters. And at 2 months, we see a ventricular-like lumen. We see a PAC6, a ventricular zone-like structure, TUJ1 expressing neurons on the outside. Here's SOX2. Here's CTIP2. We don't see SATB2 at 2 months, but at 3 to 4 months, we see CTIP2 and SATB2 in the cortical layer. Mature neurons that express MAP2AB and NUN. We see astrocytes, and we see interneurons putatively, potentially migrating from deep structures. We can make slices from these organoids, record sodium and potassium currents like Jacob Ho and Lori Isom's lab did. Kasia Glanowska, postdoc in my lab, recorded whole cell action potentials and cell-attached action potentials. The problem with the Noblick approach is the organoids are very inconsistent one to the other. They form different structures, and it's hard to compare patient and control and know that you're not just mixing apples and oranges. So other approaches have come up. Sergio Pasca, who was with Ricardo Dolmesh and now has his own lab at Stanford, came up with a cortical spheroid technique, which makes more consistent spheroids. And here we did this from a PCDH19 patient, and this is 34 days in culture, and this is 50 microns, so it's about 0.8 millimeters here. And we see PAC6 progenitors in the middle and CTIP2 cortical neurons on the outside that express beta tubulin. This gives you more consistent cortical spheroids. The problem is they don't make inhibitory neurons, so these are purely excitatory neurons. He's working on an approach where he mixes inhibitory MGE spheroids with cortical spheroids. It gets a little complicated. So we want to do this with the unaffected male mixed with control and see if they segregate differently, like Paul's mice. But there's an even better approach that Hung-Jun Song's group came up with recently that he published in Cell just a month ago. This got changed, but he called it the spin omega. I'm not sure why, but it was 3 high school students that were in his lab that did most of this. He got a cell culture. It was pretty impressive. And they ended up studying Zika virus, which is one of the reasons it got into Cell. But basically, what he was able to do, the other approaches use these big bioreactors and take a lot of media, and with the growth factors, it's really expensive. And he figured out a way with 3D computer printing to make these small bioreactors that take 2 millimeters of media, and he tested all different kinds of growth factors, and compared to the old way where you get these inconsistent, funny-looking things, you get more consistent cortical spheroids. He did hypothalamic spheroids and other regions. And they make very pretty consistent progenitor zones and then cortex. And what's really cool is he gets all 6 cortical layers in these spheroids. Reelin positive, outer layer, CUX1, SAPI2, TBR1, CTIP2, brain2. So that's the first spheroid to make all 6 cortical layers. And when they do gene expression testing, the other ones were more like 2nd trimester. These get all the way out into 3rd trimester, to 32 weeks in gene expression. So I think the field is just advancing really, really quickly, and I think we'll be able to really make hopefully nothing that's like talking and walking, but nice models to study human brain development and to look at epilepsy and how the mutations affect brain development. So I think I've showed you patient iPS cell-derived neurons should be useful for identifying precision therapy in epilepsy. The progenitors show from PCDH19 patients may show relevant phenotypes for studying disease pathogenesis. We hope to be able to combine this with animal models, although it's been proving more difficult than we thought originally. And the organoids should provide insight into brain development and may lead to therapies to restore developmental abnormalities and control seizures. And with that, I'll just acknowledge people who did the work. So Cece worked on the organoids, Kasia did some recordings from them, Yu Lu did the Dravet iPS cells, and Andrew Tidball did the SCN8A work. Helen Zhang makes all my viral vectors, and a bunch of talented physiologists in Lori's lab, and working with Miriam Meisler on the SCN8A and the Dravet project, and support from INSEAD, Perla Recherka, PCDH19, and the Cute Syndrome Foundation, and Dravet Syndrome Foundation and Cure. So thanks for your attention, and I'll take any questions.